Well, welcome back, everyone. So, just a quick recognition to my colleagues, Dr. Stock, who works with me in Massachusetts, and Dr. Victor Shen, who collaborated on all the in vivo work for this particular study. Disclosures necessary. This study that I'm about to present to you was funded by Crescent Innovations through private funding. I am the president of Crescent Innovations. I come at this technology from a standpoint of a commercial entity trying to bring therapies to market. So that's a little different than some of the academic talks that we've heard, but as long as we all know where I'm coming from, we can move forward. We've heard an awful lot about arthritis and osteoarthritis, incurable chronic form of arthritis, breakdown, cartilage of synovial joints, synovial fluid in the joints. It either results in, or depending on who you talk to, is caused by inflammation, or has some. Each one has some feedback mechanism with the other. In the U.S., approximately 12% of adults between the ages of 25 and 74 have osteoarthritis, and globally, the prevalence estimates, various respective studies have been as high as between 1 and 80%, although the majority of them fall between 5 and 25%, which sounds like about 12 to me when I do the math in my head, which is what we're at in the United States. We've had some folks talk a little bit about what the risk factors are. Obviously, I spoke earlier, over 75, 80% of all folks have it. Obesity, I know that the Framingham study and Dr. Lazowski certainly spoke to the whole issue with obesity. Women who lost an average of 10 pounds decreased their risk by up to 50%. Joint injury, jobs requiring simultaneous squatting and turning may cause up to 30% of OA and knee. We were just having this conversation offline about work-related injuries. Muscle weakness and certain ethnic characteristics and nutritional factors play a role, although perhaps not as strong, and certainly we think that genetics plays some role, but to which degree each one does, it varies from one study to another, and a risk factor is simply that, is a risk factor. It doesn't mean that you will get it. It doesn't mean you won't get it. Different types of treatments. We've heard, kind of bridging back from where we were this morning with the clinicians, certainly weight loss can play a major role in helping physical therapy, although I think we also heard that certain forms of physical therapy are a detriment. Occupational therapy, obviously, there'll be talks on that later on at this conference. Patient education, obviously, as to their behavioral characteristics to make sure they don't worsen their disease. Aerobic conditioning exercises, we certainly heard about that. I suspect that plays a lot with weight loss and obesity and the comorbidity of that with osteoarthritis. Assistive devices, footwear. And there's certainly been no shortage of pharmacological treatments. We know for a fact that non-opioid analgesics have been used, non-steroidal anti-inflammatories, even opioid analgesics for pain control, and glucosamine and chondroitin, although we did, as I mentioned earlier, and as many other folks have mentioned, we're talking much more about treating symptoms than doing actual disease modification. I'm going to speak specifically about the intra-articular therapy. I won't talk too much about corticosteroids. They seem to cause a significant amount of problems, although they do have some short-term benefits. Viscosupplementation is where I'll focus my talk. Folks have used topical analgesics to get relief from pain. And obviously, when we get right down to it, the terminal stage is joint replacement technologies. So let me talk just a little bit about viscosupplementation, because I think that this field, when I first got into it, I really liked it because to me it met what Dr. Dye was talking about this morning, the whole concept of it's really just trying to get the joint back to some sort of homeostasis. It is minimally invasive, although it's somewhat invasive. And there's certainly no shortage of FDA-approved products that are out there on the markets. But they all have one thing in common, and I think this is a problem with the viscosupplementation technologies. They're all made with hyaluronic acid or sodium hyaluronate. Sodium hyaluronate, compared to placebo, clearly has increased risks. we can see here that there's anywhere from 1 to 3 times the rate of localized joint swelling when you inject the hyaluronic acid versus saline. Why is this? Well, it's a good question. Perhaps the answer is, if we look at where the hyaluronic acids are from, in some cases they're actually tissue extracted from an avian source, which means what comes with them is contaminating proteins and some DNAs, various nucleotides. Or they're fermented via a bacterial pathogen, since Streptococcus is still the number one fermented source of hyaluronic acid. What does that mean from a standpoint of performance? Well, maybe it goes without saying, but I'm going to say it anyway, that if all those technologies are made with hyaluronic acid, they all perform, well, the same. So this is Dr. Hataro and I were having this conversation at lunch. You can see that there are 3 lines here. One line is a low molecular weight hyaluronic acid, one is a high molecular weight hyaluronic acid, and I always like to play this game. It's called Find the Placebo, because one of these lines is a placebo, despite the fact that they all show a very, very similar profile. If you were to look at any one of these in isolation, you would have a difficult time trying to tell which one is in fact the placebo. It's this one right here. And the way you can tell is it's usually the one that is higher up on the WOMAC baseline scale. So, in the event that an alternate material could be shown that was both non-inflammatory, non-immunogenic, non-cytotoxic, and was able to reduce inflammation, it may very well be that you have a new material that you can utilize and bring another tool into the clinic to hopefully treat osteoarthritis. So that's what my company has been working on. So inflammation, we've been looking at inflammation from the standpoint of matrix metalloproteinases, and in particular looking at the collagenase active group, which includes MMP-1,8, and I've kind of put a little asterisk next to MMP-13 since it's one of the few true disease-modulated collagenase active MMPs. So the way we proposed to measure inflammation was, to reduce inflammation rather, was through poly-glutamic acid. And this is the chain of it. Some of the earlier work that my company had done was in finding ways of actually manufacturing this such that it was non-inflammatory. And that actually took several years to finally develop. It's produced via fermentation using non-pathogenic bacillus, so we don't have to worry about human pathogen characteristics. We use a modified, purified through filtration buffer exchange, and then sterilize it and concentrate it. We typically, for the studies that I'm going to be showing you today, our molecular weights have been approximately 1.2 with a polydispersity of 1.1 as measured by HPLC and Mols technology. And the viscosity was measured at 4,900 centipoise on the Brookfield viscometer using a CP52 spindle, RPM 0.5. Those characteristics were chosen because that is typically how the other viscosupplements are reported. So, quick summary of preclinical safety data. That's actually easier to do than to try and do disease models. You know, we passed ISO standards, and when I say we passed, with this, our material gets sent off to a lab. They run the standards, they run their tests to ISO standards, and we have no say in that. All we get to do is to drop off the material, walk away. If there's a problem, it has to be reported back to us. But so far, we've passed ISO standards for cytotoxicity, sensitization, irritization, intracutaneous direct injections, systemic, repeated injections for toxicity, the rabbit pyrogen test, genotoxicity, and hemocompatibility, hemolysis, and implantation. Sorry, I know it's quite the soup but any technology in medical device in this material so far is considered a medical device until that time changes. These are just all the tests that we absolutely have to do before we even get to move toward a clinical solution. Oh yes, and we just found out it was listed by the FDA as generally regarded as safe within the last 2 years or something to that effect. So let me just give you a little background on this MMP study design. We had 18 rats, and they were divided into 3 groups of 6. All rats had the left knee injected with monoiodoacetate, and you can look up the details of that model. It's not my favorite model, but as I mentioned in my talk this morning, nobody seems to have a favorite model, so sometimes you have to make your choice and move forward. So 7 days later, the rats were treated with a single 50 microliter injection in that knee of either saline, Hyalgan, which is the commercially approved HA product, or our gamma-poly-glutamic acid. And at day 9, all animals were sacrificed, hind limbs were disarticulated, and the cartilage was removed. Collagenase MMPs were extracted using a 2-step procedure described by Janusz, and the results were reported as equivalent MMP1 activity per milligram of cartilage. So you could look at the concentration of collagenase active MMP equalized on a per milligram basis of the cartilage. And these are the results. So here you can see the saline group is here. Hyaluronic acid, surprisingly, or maybe not surprisingly, doesn't look too different. And this is actually something, again, we were having conversations about the marginal increases in efficacy that some of these technologies have shown. And the PGA technology is down here. And I should also add that there's a data point that I'm not showing in the HA group. It would have been about here. We had one very, very large, and we tested that sample numerous times. It kept testing out at 17. And we didn't present it on the graph because it would have made the graph difficult to read, but I'll talk a little bit more about that data point later. So, additional thoughts beyond the data that I've presented today. I want to talk just briefly about that outlier. It's possible that single injection is evidence of an adverse reaction. I did mention that all these HA technologies have a statistically significant higher adverse reaction rates than placebo. I mean, if that's really true, and I won't bore you with all the numbers, but we actually looked at what the rate for Hyalgan was. I believe I mentioned that it was 13%, and that when we figured out that given the number of animals, that having one adverse reaction in a group of 6 was within the realm of statistical possibility. I'm not sure that I will say that I'm not particularly sure that you can draw a conclusion about how a adverse reaction rate in a human compares to an adverse reaction rate in a rat. I will give you that, and I won't say otherwise, but I just wanted to have this discussion about the outlier since we are at a scientific conference and there are far better minds than I who might have thoughts on the subject matter. Now, the other piece of interesting information, and the one thing that we don't, I've never really presented it to anyone before, but it was the information about the contralateral data. The model that we used, the folks who developed the model and published it originally tested it against a group that had gone through absolutely no conditioning whatsoever, no indoacetate. And they were looking at undetectable levels of collagenase active MMPs. We decided that for the sake of the number of animals we were using and for cost reasons that we did not need to replicate a 0 number, that we would instead look at things comparatively. We did have this brilliant idea, and I say that with a slight degree of sarcasm, that we said, well, we could always just look at the contralateral limb, and that should give us our 0. Really. This was intriguing. And one of the things that was intriguing about it is that it seemed to shed, at least, maybe I'm slow to the game about an aha moment, but I don't know how many times that we've talked with patients in the various roles that I've had where it's become pretty clear that if a patient has had osteoarthritis for a very, very long time, eventually they do suffer some level of pain in the contralateral knee. In fact, Genzyme, when they reported their results from the SynVisc study, the early SynVisc studies back in the 90s, one of the things that they had concluded was that if a person had such advanced OA that they actually had reported pain in the contralateral knee, that you had less than a 50% chance of getting benefit from injecting them. On the other hand, if they didn't suffer pain or in their definition were moderately early stage, that was where they got their very good clinical results. They've always, the clinicians that I've spoken to for years, and I think this is finally changing, have said, well, the person's gait is different. They're favoring one leg over the other. It's no surprise that eventually the other leg, the knee suffers pain. Clearly, this suggests that something else is going on. I mean, if the disease-active MMPs, the only difference between the contralateral and the saline knee is that in this model, the injury, for lack of better terms, was on this knee, and this knee did not have it. Perhaps injury plus disease-active MMPs is more acute, but MMPs without injury can become a sign of chronic disease over some period of time. And I think that perhaps this is something that needs to be looked at much more so than as has been in the past. So I throw that out there to you from the aspects of science. The last couple of thoughts that I'd like to put out there, and I call this the chicken or the egg slides. Mechanistically, viscosupplementation works. It's marginally better than flushing a knee with saline. I think most folks have agreed with that. But why? Why does it work? Is it mechanical? Is it biological? Or is it other? Traditionally, if you talk, and having worked for a company that made a commercial product, Orthovisc, and I believe now their one injection is Monovisc. So I've worked with these companies, and I know what the traditional way of thinking has been. The justification for viscosupplementation has always been, it's basically a mechanical justification. It provides cushioning, it provides lubrication, and on a couple of occasions I've even heard them say, well, it replaces the degraded hyaluronic acid that's already native in the extracellular space in the cartilage. All of that's fine, except that for the most part, if you look at even in healthy human knees, material's cleared itself within 24 to 48 hours, easily. So how does that provide 52 weeks of statistically significant pain control over a placebo? It wouldn't be a scientific conference if I didn't at least share an opinion, and I will admit that it is only my opinion. When looking at degenerative joint disease, if we look at and say that this disease cycle is injury, inflammation, and joint degradation, I will give you that injury can be a very broad spectrum thing from someone's genetics to an actual ballistic accident. Inflammation, joint degeneration, and there seems to be some sort of biological feedback loop associated here. Inflammation markers can be anything from cytokines, and as I said, we're specifically looking at matrix metalloproteinases and the collagenase active ones. Regardless of what you focus on, they're all proteins. And proteins have a rather interesting physical construct, and that is that they're heterogeneously charged. They may have the molecule itself may be net negative or net positive, but they're heterogeneously charged. There's always a negative and a positive patch somewhere on that molecule. Now, hyaluronic acid and polyglutamic acid are both random coil configurations, and they're all 100% negatively charged. In the case of hyaluronic acid, it has a carboxylic acid group that is dissociated at physiological pH and conditions. One negative charge per disaccharide. Polyglutamic acid has one negative charge per amino acid. It is very possible, and in fact, I would dare say it's probable from work that we've done looking at protein interactions specifically with these charged molecules, that positive charge, that protein can orient itself so that the positive charge associates with the negative charge of the random coil, and naturally would try to shield its negative charges. That's just the way things work. If that's the case, and if the material thus clears from the synovial space, as we all know it does now, it may very well be that that's almost electrostatic scavenging, if I may use that term, to clear MMPs and or cytokines or any other inflammatory marker out of the joint space is what causes the efficacy that we see in the clinic. And the only reason we think that the PGA could be better than the hyaluronic acid for this reason is that its charge density is almost triple that of the hyaluronic acids. But one thing I will say, no matter whether you're, like I, who am trying to develop a compound, and I happen to be developing this one, and I haven't denied that, or if you're in a lab somewhere else developing some other synthetic viscosupplement, I might suggest to scientists that the mechanism of looking at how lubricating a material is might not be the correct thing to be looking at. There might be other things we need to look at, when developing new technologies. Because mechanistically, if we're looking at the wrong thing, either way, we probably will end up in a clinical dead end. So, quick status. I don't have the data. I'm not able to present the data to you today, but we have done a behavioral modification study, very similar, and have similarly interesting results. We are in the midst of preparing for human clinical trials because, as I said, and no one cares anymore about animal data in treating osteoarthritis. They want to see human clinical data. So that's, as a small little company, we're doing our fundraising to start our clinical studies, and we have plans to possibly do them somewhere in the Pacific Rim within the next year. We're probably about halfway to our fundraising goal at this point in time. And with that, I will answer any questions. I believe there is time.
Thank you very much, Prescott, for a very interesting presentation. Certainly, viscosupplementation is a very current topic in most meetings these days. Any questions, anyone?
If you speak loudly, I'll repeat the question. How's that?
No, I want to know, it's just interesting, but I want to know how much have you compared this model with what's going on with the human osteoarthritis and in relation with the cartilage, the degradation, and all processes that occurs in the development of the osteoarthritis in humans.
Sure. I personally don't love this particular model, and if I had my druthers, I probably wouldn't use it. It's a relatively inexpensive model, and at the time, my company was partnering with another company, and they more or less insisted on it. What I will say is that if you read some of the references, Kim, Janusz, and some of the others, you will see that they're able to, after certain periods of time with the MIA, they're able to see osteoarthritic lesions occur, and they can kind of grade the cartilage based on that. But I will say that there's also other papers out there that have said that it does not work that well. So that's really the best answer that I can give you. It certainly does think about what the models available are. It's some sort of inflammatory model, or it's a surgical model where you do a partial meniscectomy and look for changes. So one is mild, one is very extreme, and none of them, in my opinion, properly mimic the human disease condition.
Have a model that compares with the human. What happens with the human?
I agree, and I would like to. And as I mentioned in my talk before, I would like to have that better model.
I'll show you tomorrow afterwards.
Okay, great, thank you.
Okay, it's more complicated, but we have compared all the changes that we observe in our model with what happens in the human cartilage, mm-hmm. And when we find something in the human cartilage, we go to the model and find it also. So our model, mm-hmm. I think is more similar than other models that I've heard. And yes, it's right that we have to use a number of rats and all that. Mm-hmm. It's much work. But when you try to compare with human, you have to have a model that resembles a little bit more to what happens in the human cartilage.
Yep. I think it's very important that folks like you present those models at these conferences, because it's not just important for me to know about it. It's important for the folks higher up the food chain to know about it.
We have cooperation with some companies.
Very good. Well, I look forward to talking with you for the coffee break.
Excuse me, just ask, is any comparison with the PRP study or in this particular project? Do you compare with PRP at all?
No.
So
No,
it's
not.
not relevant?
No.
Why?
All I'm saying is we did not compare it. I don't know if it's relevant. So is that?
So you don't think it's anything to do with the biological? It's purely mechanical.
Oh no, I think it's very biological. I think the problem with the viscosupplements is that they are neither completely mechanical nor are they drug-like. I don't think that. I think that if the mode of efficacy is what I've suggested, they're not crossing a cell membrane and causing a cell to behave in a different manner. So they get lost in scientific understanding. And I think there needs to be some better independent work to elucidate these mechanisms.
Another questions around the back?
My question was kind of along that same line: is how did you choose to go this route rather than platelet-rich plasma, which is currently also being studied, or stem cell therapy? What's the rationale?
At the time, it was a collaboration with another medical device, a rather large medical device company whose name I'm not at liberty to say. That's not what you like to say at a scientific conference. But they were rather insistent on this particular model, I think because they already had some cellular technology which they also later abandoned. This is a kind of a strange thing when you get into the commercial field. One vice president changes in a company and wholesale research projects are suddenly scrapped. And if you've worked with industry, you've probably seen this happen. But that's what happened here. We retain the rights to the technology, so we get to move on to the next phase of that project without them, for better or for worse.
Sorry, coming back to the animal models which you have
Yes.
selected. Now, if you were testing any agent which is supposed to be used for as post-operative pain, then you would use an MIA model.
Mm-hmm.
But if you were going to use anything as a viscosupplementation, you have to use something which actually alters the mechanical loading, or something like destabilization of medial meniscus model, or ACL model, which actually recapitulates the osteoarthritic condition. This MIA model is absolutely of no use to be used when you're using something like a viscosupplementation, because the environment a chronic environment which MIA actually produces within the joint it actually would never give you an indication where you can evaluate how your viscosupplementation is working. And that too, when you're going from small animal models to the medium size or larger animal models and then moving into humans, of course, this correlation will further become the gap of your preclinical studies going into clinical trials is going to expand a lot more.
Yep.
So there's a lot more variable where you're starting from. Now, I don't know if you're able to disclose it. When you're going into the clinical trials, what kind of patients you are selecting?
So let me comment on your comment first, if I may. Yes, I agree. It's not the best model. The ironic thing is that we've now worked with 3 global manufacturers who all have in their portfolio viscosupplementation products, and they may not use the MIA model, but they all use some sort of chemically induced pain model, and that is their value. That is the method by which they evaluate it. Is it the right way? Probably not. I don't think it is. Now, remind me.
So you're saying to me the results are not presenting the real story then?
Well, they present something. They present something about inflammation. They present some. As I mentioned, we've done a behavioral study looking at actual pain on incapacitance scales and whatnot, and there's clearly data there. Is it? I have not claimed to you that this is somehow a disease-modifying therapy, sure. So that would clearly be out of line. But I don't know if you were here for my little 5-minute spiel at the previous. When I said that the biggest problem that I have as a company developing technologies to move through the commercialization pipeline is that there is no well agreed upon animal model by which these studies can be done. It's a big problem for us, and as I said, it's a log jam that not just my company but several other small innovative companies are all running into this. The big companies don't want to bring the technology up, and it's difficult to get funding and financing to move these projects forward. So it's a challenge, and it's something that I clearly have to work with. Now, what was your second?
So in terms of selecting patients for the clinical trials,
Yes.
I don't know if you're able to disclose that. So how are you actually selecting patients for this particular study based on the results, what you have from your preclinical?
Well, so far we're, again, may not be perfect, but we've looked at, what the selection criteria have been for some of these other early, commercially viable products. We're not looking for people with highly advanced stages of chronic forms of osteoarthritis. We're looking at early stage. And I guess when you think about it, if you think of, kind of take a step back and look at this from the bigger picture, if you will, it's almost not surprising why some of the big companies that have commercialized these technologies perhaps have gone this route. If the model is essentially showing you just an early stage inflammation without necessarily severe joint degradation, wouldn't that be the patient population you would look for? You're trying to provide alleviation to the patient population that's early stage, but perhaps hasn't gone to the point where Dr. Dyer was showing us almost complete joint failure. If we can do better studies in a surgical model that shows us how the joint itself changes over long periods of time as we provide various types of therapies, you can derive from that a better targeted patient population, in my opinion. I think that ultimately is why we need to look at better models in the future.
Okay, I have a little basic question that why you use polyglutamic acid?
Yes.
Did you try polyamines? Because that might modulate your environment, protein environment at the same time, like spormidine or spormine.
The answer is we didn't, so we don't have any. So I can't. So.
And then did you try to look at the matrix metalloproteinase inhibitors? They are also playing very important role. All the inhibitors for matrix metalloproteinases. Because the balance of your degradation versus formation is the major things for good joint health. So when you treat with polyglutamic acid, what happens to the matrix metalloproteinase inhibitors?
So we did not look at that, and part of the reason why we didn't is because we didn't want to. We only had 3 groups, and as I said, there's only so much we can do with the funding that we have available. What I would say is that the other issue that all these studies eventually may support an FDA submission at some point in time, and you don't want to be throwing extra levels of complexity into that. We wanted to kind of keep this as a kind of a straightforward biologically inert molecule rather than looking at some of those other things. But if you go to some of the literature, which we have looked at very carefully, there's an awful lot of folks who are looking at various types of agonists and whatever of MMPs, MMP inhibitors that was certainly looked at for quite some time. The issue with them typically is that the paradigm in industry, when you're looking at an MMP inhibitor, for example, that's being developed by drug companies typically. So a drug company takes this paradigm that you take a pill and you get some sort of systemic reaction. Well, typically the side effects of that systemic reaction are worse than the benefits that you get at the local level. So
Thank you.
I'm not sure that it really answers your question, but
No.
it gives you what our thought was when we designed the study.
Thank you so much.
Oh, you're welcome.
Any other questions, anyone? Okay, thank you very much.
Thank you very much.